au Zoom. Pour les gens sur Zoom, Jonathan. Ça, c'est pour les gens sur Zoom.
Non, j'ai fait un genre de montage. J'ai fait un montage. Cet ordi-là est connecté à la réunion Zoom et il envoie les images.
Ok,
Celui-là réceptionne les images et YouTube enregistre sur celui-là.
ok, ok. Puis le prélance et après tu donnes. Tu le mets. Bon. Ce qui va passer le diapo.
Right, as I say, we're running about an hour and a quarter late already, so we'll have to make sure we keep to time. So the first up, as almost usual, is Chema, Professor Guiterres. So thank you, and we look forward to hearing on another presentation from you.
You have the end. You can do it.
I can do it. Well, good afternoon, everybody, and thank you for the opportunity to present this work on. I will present a reflection on strategies to improve the availability and accessibility of antivenoms in the world. Okay, there you go. So we have seen this map before. So basically, snakebite envenoming is a big public health problem in the world. We don't need to talk much about that because we are convinced. So the WHO issue a global strategy to control and prevent snakebite envenoming. And this strategy is based on 4 pillars: empower and engage communities, strengthen health systems, increase partnerships, coordination, and resources. But a very important pillar is to ensure safe, effective treatments. And we know that the only scientifically validated treatment is the parenteral administration of antivenoms. So we need to have in the world, as general, enough volume of antivenom. We need to distribute this antivenom and to use it correctly. And this poses many challenges for our community. This map shows the universe of antivenom manufacturers in the world. So we have a good group of antivenom manufacturers in Latin America, some in Europe, in Asia, Australia, and a few in Africa. But this universe is quite heterogeneous. There are some antivenom manufacturers which produce only for the national needs. These are mainly public laboratories in the global south. Other manufacturers are regional producers. They produce antivenoms for their own country, but they also export antivenoms to the region. For example, the antivenom manufacturer in Thailand. And then a few manufacturers are global producers, which produce antivenoms for different regions and different countries. So it's a very heterogeneous collage of manufacturers. And there are differences in terms of volume of production. In some cases, they produce 100,000 vials per year. In some cases, they produce 10,000 vials per year. There are differences in the technological platforms. Some producers have very well-developed technological platforms. Other producers have weaker platforms. There are differences in the national and regional scenarios and differences in the needs that they have to fulfill their roles. So this is a very heterogeneous universe that needs to be strengthened. In particular, of concern is the very limited number of manufacturers in Africa. There is a manufacturer, a traditional manufacturer in South Africa, and there are some manufacturers in the northern, in the Maghreb region of Africa. But in sub-Saharan Africa, there are no antivenom manufacturers, and they rely on antivenom products from other regions. So the first need, I will speak of 3 tasks to improve antivenom availability and accessibility. The first one is to improve the production of existing laboratories. So we need to support laboratories that already produce antivenoms that have difficulties, see how can they be strengthened. They need to upgrade the technological platforms to implement good manufacturing practices. They need to improve the qualification of the staff, and they need to incorporate processes of technological development and innovation. This goes particularly for public health laboratories. And here in Latin America and the Caribbean, we have this situation. How can these laboratories can be improve their work? Well, like I mentioned this morning, the networking and international collaboration is very important. Workshops, training, technology transfer. There needs to be investment in infrastructure, technology, and personnel, and this, particularly for public laboratories, depends on the support from the governments, and this support is not always present. And then there is a need to implement the WHO guidelines. So the strengthening of antivenom production laboratories that currently produce antivenoms is one scenario that we need to promote. And a good example is this RELAPA, this network of public laboratories in Latin America, which is working on the strengthening our local capacity to increase the production and the quality of antivenoms. We hope that in the next years, our continent will be in a much better position in this regard. The second scenario to improve availability and accessibility is one that has been explored little, to a lower extent, and is the technology transfer. So laboratories that have the know-how to produce antivenoms need to transfer this know-how to other countries or laboratories that need to start production of antivenom. So we need to identify the possibility based on endogenous technological and institutional conditions. And I will give you one example in which we in Costa Rica are involved. Several years ago, we were contacted by colleagues from Sri Lanka. Sri Lanka is a country that has 30,000 bites per year. And they rely mostly on antivenoms produced in India. But they wanted to produce their own antivenoms. So we started a technology transfer project involving our university, the University of Peradeniya in Sri Lanka, and a non-governmental organization of people from Sri Lanka living in the United States called Animal Venom Research International. So with this partnership, we in Costa Rica, we got the venoms from Sri Lanka. We in Costa Rica developed a pilot batch of antivenom, and this antivenom was tested preclinically and was sent to Sri Lanka. Is going through a clinical trial right now. I think it's almost finished. And the idea would be not to produce this antivenom in Costa Rica, but to transfer the technology. However, Sri Lanka has faced many political and economical problems, and the funding for this technology transfer has not appeared yet. But this is a possibility, and it's an interesting example of technology transfer. There are some initiatives in sub-Saharan Africa, in Nigeria, in Kenya, at least in those two countries, to develop a local production of antivenom with the collaboration of laboratories, other laboratories, including Micropharm, which is involved in collaboration with Nigeria. And recently, the World Health Organization is promoting also collaboration with Cameroon to develop their own antivenom. So the transfer of technology is a possibility to improve the availability of antivenoms. And the third scenario, which is working, is the commitment of laboratories that currently produce antivenom to produce antivenoms for other regions and countries. And this third scenario involves laboratories from Mexico, Costa Rica, Brazil, the UK, South Africa, India, and Australia. There are laboratories in this country that produce antivenoms and deploy these antivenoms to other settings. For example, in Africa, Micropharm from the UK is providing antivenoms. Our institute is providing an antivenom, and laboratories in India are also providing antivenoms. So in this way, laboratories with good facilities, with production capacity, can contribute to the availability of antivenoms in other countries. And I would like to mention one particular example for which we are very proud, and is the development of an antivenom for sub-Saharan Africa. A partnership was developed early in the first years of the 2000 decade between the Federal Ministry of Health of Nigeria, the Liverpool School of Tropical Medicine, Micropharm in the UK, the University of Oxford, and our institute in Costa Rica to develop an antivenom or antivenoms for sub-Saharan Africa. Micropharm developed an antivenom called Equitab G, specific for Equus ocellatus, which is the most important snake in the western sub-Saharan Africa. And in Costa Rica, we developed a polyvalent, polyspecific antivenom also to be used in western sub-Saharan Africa called Equitab Plus ICP. So to develop a new antivenom for another region, I will mention the steps to be taken. Well, here is showing, this is the antivenom that we manufacture for sub-Saharan Africa. It's called Equitab Plus ICP. This is the paper where we describe the development of this antivenom. And this antivenom originally was developed with the venoms of 3 snakes. And now, with the work of the industrial division of our institute, recently we published this work whereby we expanded the neutralizing capacity of this antivenom by using a combination of 22 venoms. Instead of 3, 22, that would cover most of the venomous snakes of Africa. It worked very well preclinically, and we want to pursue clinical tests with this antivenom. So the first step when you develop a new antivenom is to select which venoms are you going to use. And for this, you need epidemiological information, clinical information, and information on the cross-neutralization of the venoms and the antivenoms. So with the knowledge that had been gained in sub-Saharan Africa, the selection of the venom was based on which snakes were more important, which snakes caused the most accidents, and what would be the cross-neutralization of these venoms. So initially, we started with the venoms of these 3 snakes, Equus ocellatus, called carpet viper, Vitis arietans, the puff adder, and Naja nigriculis, the spitting cobra. So with these 3 venoms, we started manufacturing the Equitab Plus ICP. Also, we did a project, I will not talk much about that because of lack of time, but we did a project with Papua New Guinea, in which we prepared a monospecific antivenom against the venom of Oxyuranus scutellatus, the taipan, one of the most venomous snakes in the world. And this antivenom worked very well preclinically. It was tested clinically, but for a number of reasons, the production did not continue. So once you select the venoms that you need to use, then the next step is the preparation of pilot batches of antivenom. So in our case, we prepare a pilot batch of this Equitab Plus ICP, and we characterize this by preclinical testing and so on. Recently, we developed at the experimental level another antivenom for the Middle East and Northern Africa called Menavip ICP, which underwent preclinical testing, and we expect to have some clinical testing. And this is the paper where we describe the antivenom for Papua New Guinea. So you need to prepare pilot batches of antivenom, and then you need to test these pilot batches preclinically. So in this case, I show the results of the study we did with the 3 venoms we used for Africa, Equus ocellatus, Vitis arietans, Naja nigriculis, showing that the antivenom was effective in the neutralization of lethality, hemorrhagic mytotoxin, thermonecrotic, coagulant, and the fibrinogenating activity. So preclinically, it was well evaluated. It had good neutralizing capacity. And the next step was to test this antivenom not only against the 3 venoms used for immunization, but also against other venoms from Vitis and Equus in Africa. And the antivenom proved to be effective in the neutralization of these other venoms, not using the immunization, but having close immunological relationship with the venoms used for immunization. Also, in the preclinical tests, the work of Juan Calvete in Valencia and other groups introduced this assay or this method of antivenomic analysis. I don't have the time to go in the details, but basically it's an affinity chromatography method that allows you to detect which toxins in a particular venom are being recognized by the antivenom. And here, basically, we show this is the HPLC profile of Vitis gabonica venom, and this is the profile of the venom after it is passed through a column where we have antibodies against this. And you see most of the bands, most of the peaks are depleted from the venom, showing that the antivenom has good antibodies against this venom. So this is another tool that can be used in the preclinical tests. Once an antivenom has shown to be effective preclinically, then we need to move to preclinical tests. But before that, I like to mention that, like yesterday I mentioned this, the World Health Organization is developing a risk-benefit assessment of antivenoms to determine objectively and rigorously which antivenoms are safe and effective to be used in different settings. Right now, they are working in Africa and Asian antivenoms. So they evaluate a dossier. The manufacturer has to present a dossier. They do laboratory analysis in terms of chemical, physical, biological tests. They do preclinical neutralization efficacy, and then they visit the laboratories to ensure that they fulfill with the good manufacturing practices. And if the laboratory complies with all these requirements, then this antivenom is placed in the list of antivenoms recommended by the WHO. So this is a very positive step forward in our field. So I was saying that after the preclinical tests, ideally, after the preclinical test, ideally you should go to clinical assessment of antivenom efficacy. This was done for our antivenom in Nigeria. Then you need to register the antivenom in the countries where it will be distributed. And then it's important to do pharmacovigilance, to follow up on the performance of the antivenom when it's used in large-scale conditions in patients. There is also the issue of distribution and accessibility. This goes with the public health policies of the different countries. And therefore, in summarizing, in order to improve the accessibility, availability of antivenom, we need to have an integrated intersectorial and interprogrammatic strategy, including the scientific community, the manufacturers of antivenom, the national public health authorities and local health communities, the international partners that promote access of antivenoms, the international and regional health offices, and the organizations of the civil society, working in a cooperative way. So some of the data I presented have been done in collaboration with all of these groups from different countries and continents. And I think the cooperation is the way to go in this field of antivenoms. So thank you very much for your attention.
Thank you, Chema. And we'll save any questions till to the end of the session, just given we're running short of time. So can I invite Aaron to give his presentation? Please, Aaron. And I'll keep with your time as well.
Not clicker. Because with Zoom you cannot use the clicker. The Zoom sharing blocks the clicker. You will have to. Sorry? It's okay. But you don't have to ask for. Huh?
Ready? Okay. Good afternoon, guys, and thank you again for the invitation. Since we are been talking about all these, you guys hear me now? Yeah. Since we have been talking about all these venom availability, accessibility, I will try to get not detour from the path that Dr. Gutierrez has been talking about. So I will just came up with this presentation title. Is pretty much the role of the ICP in the Caribbean scene, since we are the new player in town. So some topics that I would like to discuss with you guys, mainly focusing on the venom availability. How about the market? This is kind of just trying to take you guys a different picture. Again, same in the same scenario of all these scientific collaboration that we need, but we are going to discuss some of the product characterizations. So we have two main, let's say, well-established products in the region. Well, we can say three, but two more well-established. And some other things, let's just as a take-home message. So this is kind of the same table that we have seen it since yesterday, I think. So we have some availability of antivenoms. Sorry, I just launched for the Caribbean region itself. So you can see some areas. I will try to use this. Some areas that we cannot, but if you see the species of the snakes that are going to be that, the species that are present and distributed in the places as Belize in the Central America or some other places like Suriname, you will see that there's probably some different sources of polyvalent antivenom that can be covered by the Clodomiro Picado Institute. Just to start off with the Clodomiro Picado Institute, the Polyval ICP antivenom is a polyvalent based on now 3 main genus, which is the botropic, which is all the fertilances, the crotalic one, which is the rattlesnake. So we can cover some rattlesnakes from here. Where is it? From here. And we also have the lachetic component, which is the bushmaster. So we are now talking about the polyvalent, polyspecific antivenom, compared to others that just focusing on some species and some genus, which are just making them monospecific, monovalent. So this is the current situation of the antivenoms in the region. So we have, let's say, at least 12 different antivenoms that we can use in the Americas, including the Caribbean. But this is the things that I want to stress out in the presentation, and as we all have been talking about this, the challenge of the snakebite antivenom viability, and this is the concept or the question that I want to give you guys to think of, is if we have a very, very good system to provide antivenoms, but the difference will be the system or the methods or the pathway that we have to access the antivenom. So most of them, and we have been discussing about research and development, we are considering neglected disease as the whole umbrella that is covering the problem of the snake bite. But this is one of the things that since yesterday we have been focusing on, which is the limited viability. Where is my mouse? So remote areas and impoverished regions. This morning, this snake session morning was huge success, guys. Was talking about how far are the patients from one point to just get the first aid and receive the antivenom. So this is one of the things. And the other thing that we need to focus on is on the public health programs, especially in the primary health cares. So how easily could be just to get access to the antivenom. So one thing is having a lot, quoting a lot of antivenom. So how about the offer? Are we well covered by antivenoms? And the other thing is how we can access to use the antivenoms in the correct way. The other thing is what we need to train people to apply correctly the antivenoms. So are we well covered by antivenoms? Are the Caribbean region well covered by antivenoms? Since top of my mind, we can say that we have at least 2 main products that we can explore and work with. But is the access to this antivenom well enough covered in the region to be a very specific point to address in future discussions? So antivenoms for the Caribbean, of course, we have Botrofab is fabricated, is produced against Botrops lanceolatus, the Martinique fertilans. Then we have the Polyval ICP, which is those 2 will be the focus of my next slides. Then we have Activimim for the French Guiana, which is interesting. They have, let's say, not that the best results to work with the antivimim. And sorry, that's Botrops actros. And the antivimim is a nice quoting mix of immunogens. So again, we are talking about Botrops lanceolatus as one of the major players in the scene. And then we have talking about Polyval ICP as antivenom. So we have 2 different formulations of antivenom working real good, with different, let's say, targets, venoms, or snake species. The same at the end of the day is try to save lives, and they both work very well in different, let's say, manners because of the fabrication, because of the production of the antivenom. So we went out and tried to see all these challenges in the antivenom availability. Again, most of the thing is we can have a good, let's say, good networks of antivenom. As Dr. Gutierrez just mentioned, we can have pretty much 20 to 23, almost 32 producer centers. But the thing is, how we can take those antivenoms to the specific region. So supply chain is a challenge. The lack of awareness is kind of an interesting point to think of, because we need to put attention on the places. As we were discussing in this morning in the other session, there's, at least in San Lucia, there's two main specific points that will need a specific attention in terms of having the antivenom locally. And we are discussing at some point the insufficient production of the antivenom, but definitely economic barriers, since this is a neglected tropical disease that tends to be impoverished people or countries. So we need to address this as a whole. This is being the most of the thematics that we have been talking about. So considering this, we just moved to try to analyze the market in the region. So we have, for Martinique, we have Botrofab, and for Botrofab is against made specifically for Botrops lanceolatus, but again, we have another new player, if I can say so, which is the Polyval ICP, and we would like to call Caribs. That's my French, sorry. But again, in French Guyana, we will have antivibmin against Atrox lanceolatus, among others. And again, we will have the Polyval ICP Caribs. This is if you guys see, is a trend to have different antivenoms, multiple antivenoms to cover pretty much the same species. In this case, Botrops. And in San Lucia, we have Polyval ICP, which is a nice, excellent cross immunorecognition, as Dr. Gutierrez mentioned. So we can cover the whole Botrops genus with the regular traditional fabrication, including the mix of immunogens that we have in our immunization scheme. So San Lucia is well covered, actually excellent cover, and the news from this morning session was since 2017 we don't have fatalities. So it's just saying that the antivenom is working real well. So we did a bit of product characterization. So we took the Botrofab, and we will see that this is just to showing that it's a different way to fabricate, to produce the antivenom. This is a FAB2 fragment. We don't have the time to go with that, but we can talk about later. So you see that there's some difference between, you can see compared to the Polyval ICP Carib. So we will do protein content, which is kind of protein content, and the effective dose, which is the neutralization of the lethality. Those are let's say not the major, but they are very important things to look at when you are characterizing an antivenom. So you see that they have some nice numbers there, and actually they have a very interesting amount of venom that can be neutralized. So we have some differences. We analyze at least two batches, and for the Polyval ICP Caribs, we analyzed 3 batches that include the Caribbean venom component. I will talk about that later. Sorry for this. So we did this, and this applied. Sorry, see, I'm running late. We applied some of the lethality of neutralization just to characterize some of the schemes and strategies that we use to produce the Polyval ICP Caribs. Then the other talk I will talk about this, and we have some data before. So we have Asper here, and then we have Lanceolatus as neutralized. So we have some data, and then we have some good results again with the immunization of the Caribbean venom component. And most of the tables you see are reference. So the thing is, we have 2 products which are different, but doing at the end in the clinical aspect or in the hospital the same effect, which is saving lives. So we have different pharmaceutical formulations, whole IgG, which is the ICP, Polyval ICP, Caribs, And then we have the fragment AB2, which is the Botrofab, which is the other antivenom that they are using in Martinique. Based on that, we have different pharmacokinetics, so the way that they can behave in the clinical aspect and for the physicians is different. So you need to consider that they have different, this is for the pharmaceutical pharmacists and the clinicians, they have different clearance times, they have different median lifetimes when the antivenom is applied. So we need to consider that. And this is the interrogation because different neutralization effectiveness is that a real question? It's pretty much not a real question. It's the way they are doing the neutralization of the main effect, which is just to prevent deaths. And both of them, if you just go through the retrospectively, if you go with some of the works doing with the Botrofag, with the Botrops caribeus antivenom, you will see good immunorecognition, cross immunorecognition, as well for the polyvalent ICP. So for the polyvalent, we will have a good and a wide species coverage range, which is just make it very, let's say, easy to have access to different species, which is just some of the, let's say, advantage of that. But at the end, and depending on the, as I was talking about, depending on the pharmacokinetics, most of these things, and remember that the question that they just started off was access to the antivenom instead of viability of the antivenom will reduce to price. Are we willing to pay more for some products, or that's the decisions that all these stakeholders has to make? So my take-home message for you guys is variables such as socioeconomic factors, vulnerable populations, challenge in accessing healthcare systems and specific or effective antivenom, which is both of them are very effective. Enhancing snakebite treatment capacity in primary care systems and advocating for increased funding and policy changes because, again, price is a factor that we need to consider. Will impact the continuous antivenom fabrication and its accessibility. Considering that, we need to reconsider and rethink if the market is well covered by antivenoms, how easily will be to have access to the antivenom. And all this work is just made by all these people, the ICP antivenom team and all the sponsors. Thank you, guys.
Now we have Adam Galel. I think, Jonathan, you're going to get him online, yeah?
Atem, tu nous entends? Attends, on connecte. Oui.
Whilst we're waiting for Jonathan, has anybody got any questions for Aaron or Jonathan? There's one question there.
Ok, tu peux mettre en plein écran.
For which reason would you like to identify the venom? Once you get the venom from the snake or the patient?
Yeah, for the patient. I heard you were mentioning something with markers, but I wasn't sure exactly what that was. Oh.
Good question actually, because the ICP has a group developing multilateral flow testing, and there's people in Denmark. So they are developing these portable tests. Say, so you can take either urine or blood samples and put it in like the COVID test. Remember that with the old brands, so you can put the sample there and start running the study and see if there is, for case or example, will be either fertilized or maybe. So for the Denmark group, they are working with the Jaralakas, which is the fertilized from Jaralakas for the seal, and they are also looking for the bushmasters in the seal. Because sometimes the pathological physiological effects can be confusing. So they need to identify which venom, which species was the causing affecting, to provide the correct antivenom.
Okay,
And
Aaron,
I think,
Professor is ready. Very much.
Allô, professeur Kalel. Oui, très doucement. Est-ce que tu peux partager ton écran à nouveau? Ok, c'est bon. Essaye de parler un peu fort.
So it's okay? I can start?
Yes.
Okay. So, good afternoon, ladies and gentlemen, my dear colleagues, and sorry again for not being here physically. In this talk, I will speak about snakebite envenoming and access to antivenom in French Guiana. So, as I said before, in French Guiana, there is almost 100 snakebite envenoming per year, 35 of which are managed in Cayenne Hospital. And half of them attended first the remote health care facilities, and the culprit snake is Bothrops atrox in most of cases. Since 2017, antivenom was introduced in French Guiana, and the molecule used is antivitamin 3. It is used under a compassionate use regarding to the French law and regarding to the food and drugs association in France. In 2020, we published an article with Gabor Rizier regarding the efficacy of antivitamin 3 in treating snake-bitten patients in French Guiana. We studied 133 patients, and we found that recovery from coagulopathy was earlier with antivenom than in the control group without antivenom. Without antivenom, fibrinogen recovered in 63 hours, while with antivenom is recovered in 30 hours. In this study, the time to antivenom was 9 hours in median, so it was late. And this is explained by the availability of antivenom only in the 3 main hospitals, as I explained before, and patient took time to be transferred from remote health care centers to the main hospital in Cayenne. And sometimes they walk through the forest or they travel with boat to reach the nearest healthcare center to be transferred after that. In 2023, we published an article in PLOS Neglected Tropical Disease to compare patients who received antivenom early, before 6 hours, and those who received delayed antivenom more than 6 hours. So, and we found that the early group
Allô, Atem? Tu peux reprendre 2 diapos plus tôt, s'il te plaît?
Yeah?
Vas-y, c'est bon.
It's okay? It's okay?
Yes. Yes.
Okay, so I explained that antivenom is available only on the hospitals which are in the coastal side of the department, and that patients who present to the remote healthcare center must be transferred to the hospital, mainly in Cayenne, to receive antivenom, and this takes time. The transfer is by helicopter, and one-way travel will take between 1 hour and 1 hour and a half. So we studied the efficacy of the antivenom on the recovery of coagulation parameters in patients who receive early antivenom and those who receive delayed antivenom. So this is why patients arrive tardy to the hospital, because they have to travel through the forest or through the rivers, and this is the helicopter that we use to transfer patients. The result of this study show that in the panel A, that the time from snake bite to normal clotting was shorter in patients who receive early antivenom, than in those who receive late antivenom. However, the time from antivenom administration to normal clotting factor is similar between the two groups. So we drawn this hypothetical schema where we explain that, so this is the kinetic of the venom. When patients receive early antivenom, fibrinogen or clotting parameters are decreasing, so that when we give early antivenom, they can correct the disturbance. However, when we administer delayed antivenom, the parameter coagulations are already recovering, and delayed antivenom will not help people to recover. So at the same level of low fibrinogen concentration, we can be in the decreasing part when we are early from the snake bite, or at the increasing part of the coagulation parameters synthesis. And so that the efficacy of the antivenom is not similar. However, in some case reports, very late antivenom administration was shown to effectively reverse the toxic syndrome. There is 4 reports in the literature about this. So that we investigated patients who received late antivenom between 6 hours and 12 hours to patients who receive very late antivenom, which we defined more than 12 hours after the snake bite. And we compared this group to patients who didn't receive antivenom. Those patients were involved at the time when we had not antivenom, or in some shorter periods. And we found that patients who receive late antivenom between 6 and 12 hours recover their fibrinogen earlier than those who receive very late antivenom, which group it progress exactly similar to patients who didn't receive antivenom. And in panel B, when we study the ratio of patients with normal fibrinogen dosage according to the time from antivenom administration, we found that very late antivenom recover earlier than late antivenom. So same things we found with INR, but no differences in the time to recover from antivenom administration, and something with EPTT. So we concluded that regarding to the first study published in 2023, that patients who receive early antivenom recover earlier than the other patients. Patients which receive late antivenom between 6 and 12 hours benefit from the antivenom administration, and antivenom will accelerate the recovery of coagulation parameter. However, patients who receive very late antivenom, more than 12 hours, they progress, they have exactly the same outcome to those who didn't receive antivenom. And with regard to the 4 case reports I showed you before, we drawn this hypothetical schema where we study the coagulation parameters in blue and the venom kinetic in red. And we suppose that the venom can be either a short half-life venom, so that in the very late phase, the kinetic of the venom is too low and the coagulation parameters are already recovering, so that antivenom may be not effective. However, in case of venom with high half-life, even in the very late phase, there is still circulating venom, and the coagulation process is still involved, even in the very late phase. That's why probably in the 4 case reports that I showed you, this was administering antivenom was benefic in the very late phase. So in conclusion, it is obvious that we have to provide remote healthcare centers with antivenom. We have to train teams. We have to search for more effective and less expensive antivenom. We have to develop specific antivenom for French Guiana, and we have to better understand the pharmacokinetic or toxicokinetic and pharmacodynamic of the antivenom. Thank you very much.
Thank you very much, Professor Atel-Kalel, for your concise, clear presentation about time to antivenom. It's time now to receive our colleague, Ian Cameron from the UK, from Nipro Farm, for his talk. Thank you. Will give us an insight into the manufacture of both of our, that's the antivenom we use in Martinique. Thank you. Do you have a short question for Mr. Khaled? No, sorry. Okay. Short question for the previous presentators? No? Are you sleeping? Coffee? Wait.
Yes. C'est le lien Zoom qui... Oh, ok, sans Zoom. Okay, we do it like that. Okay.
So what's the, just down, yeah? Right, well, thank you very much, firstly to Davor for inviting me, and thank you to the organisers who put in a lot of work to make this a very successful couple of days. I also like to take the opportunity to, just acknowledge the work that ICP has done in the antivenom world. I often get calls from clinicians in Africa saying they need an antivenom, and I always know if I send them to ICP that, that's right. I can always send them to ICP and they'll help. So I think ICP is known really well throughout Africa as a sort of reputable manufacturer of antivenoms, which really save thousands of lives every year. So we're a very different organisation. It's a privately held company. In terms of, right, sorry, wrong one. In terms of declaration of interest, I'm a director of Micropharm, a small shareholder, so I'm not the owner, so don't get excited. Also, I think I've been the CEO of Micropharm for 10 years now, but I was a non-executive director of Micropharm for 10 years before that. I was working in finance in London, doing investments in often a number of life science companies, but the opportunity came to take over from Professor Landon as a CEO, and since I was 50, I thought if I don't actually run a company, I'm getting too old to do it. So that's why I made the move to actually working as CEO in the company. So, there's what we do. Our history is in antivenoms. Already Chem has mentioned the project with the Nigerians and ICP on Equitab G. we've been supplying Equitab G to Nigeria since 2002, so we've worked there a long time. We operate within the UK, and our regulatory authority is the UK Medicine MHRA. They're our sort of statutory body that ensures we comply with GMP. We have 2 manufacturing sites. We've got the original one, which we built for the Equitab G project, and then we built a new facility to manufacture the Sanofi products, which we acquired from Sanofi in 2017. There's 75 of us in the company. I mentioned the split between production and quality because the regulatory burden and scrutiny we are subject to under the sort of UK or EMA regulatory authorities is very heavy. So we need a lot of people to just making the antivenoms in some way the easiest bit of the job. Making it comply with GMP and keep the regulators happy consumes more time and resources. So I've mentioned Equitab G. Professor Gutierrez will comment on whether it's Romani or Ocellatus. I think there's a debate. It's been going so long that they've changed the classification as we've gone through. There's Viperfav, which is the European antivenom, so we supply all of Europe. And then our special product, Bothrofav, which we've talked about a lot. Because of the relationship with Sanofi, we're also going to reintroduce Favafrique next year, and after in 2026, Favirep. Favirep is for the Middle East and North Africa. Both were successful products for Sanofi in the early days, but the lack of interest in those products, both from the market and the owner, they were discontinued around 2014. But the Wellcome Trust in the UK has helped support Micropharm to redevelop the manufacturing process for Favafrique to meet current GMP. We're also a bit unusual in the antivenom world. We've diversified, and because as my chairman keeps telling me, if you want to be rich, don't get involved in antivenoms. So we've used our sort of technology and know-how. We've been working with the UK Health Security Agency for the past 10 years. We're using the polyclonal platform to develop antibodies to the toxins A and B of C. diff. And next year we'll be starting a clinical trial, phase 2 clinical trial with a site in Nairobi and a site in Perth, Australia. So we hope by about 2027 we'll have data from that particular trial. It's different in that it's an oral product. So because the toxins are in the gut, there's no point giving it IV. So if you ingest the antibodies, they'll neutralize the toxins that are present in the gut. In terms of the manufacturing process, as I said, our original manufacturing plant where we manufactured Equitab G and our European antivenom Vipretab was not large enough to facilitate the inclusion of the Sanofi manufacturing process. It's a lot more complex than what we had in our own facility. So from 2017 to 2024, we transferred the technology from Sanofi and we built a new manufacturing facility to manufacture those products. So it's been mentioned before, but very briefly, I'm sure you're all aware, the key is to immunize the horses, although our Equitab G product is actually we immunize sheep. You actually take the serum from the animals or the plasma, and that contains the antibodies which are then used, which are then purified in the process. And there we've got a chart at the bottom or flow diagram, but there's actually a bit more to it. So in the manufacturing process, we transferred from Sanofi. You pool your plasmas together, you add solutions, you precipitate and you centrifuge out the euglobulins, they're called. We then apply chromatography to capture the antibodies, which we're going to then purify. We then cleave the antibodies. So we've talked about IgG, but this is a FAB2 product. So we do pepsin hydrolysis, which splits the FAB2 from the FC fragment. We then do another chromatography step to then purify that FAB. We then pasteurize the product. Now, people say you shouldn't heat antibodies, but as part of the viral clearance, because the regulator is very concerned about transmitting viruses from animals into humans. So we actually pasteurize the product to ensure that we kill any viruses that may be present from the host animal. We then purify again the bulk active substance. We formulate it in a final formulation, and then we do the filling and the packaging. So it's a multi-step process. Every step is, we only operate sort of 12 hours a day, so the process is broken up into a number of easy sort of slots. So about the clean rooms where we actually make the product, they're Class C, so that's quite a high grade of clean room. We have Class A laminar flow units, so when we do manipulations to avoid getting bacteria contaminating the product, this is done under those areas. We also segregate the area between the pre-viral, so when the plasma is there, it may contain viruses, and then once we've pasteurized it, it's in another part of the facility, so that there's no risk of transferring viruses across the facility. We also then obviously have all the necessary laboratories, clean rooms, and the like. So just a few sort of pictures always to brighten up a presentation rather than just me nattering on. So you can see here to the left and the right, we've got the centrifuges. We use disposable technology. They're closer. Thank you, Dabor. So we use disposable technology, which means actually we process everything in bags rather than in steel tanks, which then have to be cleaned. We then, another shot of the clean rooms where in the foreground you've got the chromatography systems, which we use for the purification. The pasteurization oven, so to, again, ensure segregation, you put the tank containing the FAB2 into that oven, and then you go into another room and you take the oven out, the tank out from the other side, ensuring segregation. That's in the post-viral area with a laminar airflow unit where we do final formulation. And then we go back to our original facility where we ampule fill the product. We use ampules because we found in Nigeria there's a lot of fraudulent use of used vials. So there are reports and continue to have reports, especially of the Costa Rican product, where people are getting the empty vials, filling them with water, and then selling them to unscrupulous people. So with an ampule, that can't happen. So as I say, I mentioned disposable technology. It's easier from a cleaning point of view, but it produces a lot of plastic waste and increases our manufacturing costs. So a bill of materials to make one batch comprises over 120 different items, and we use a lot of solutions because of the chromatography. So for one batch of Bothrofav, we're using over 54,000 litres of solutions and water to manufacture one batch. And if you think 1 litre of solution costs more than a pint of beer, you can imagine the costs involved. So just for the scientists amongst you, I know Aaron may be interested in this, but we start off with 200 litres of plasma, and we end up with 1.5 litres of released product. So that's the sort of huge sort of purification step. If we want to look at the actual proteins, we start off with 4 kilograms of IgG, or immunoglobulin G, which contains the antibodies that are going to be binding to the toxins, and we end up with 325 grams after that purification. So you can see the extent. Every time you do a manipulation, you lose product. Now, one of the factors in that is also the time. So if you look at the timelines there, you're talking of basically 6 months from the start of a batch to actually releasing the product. So, people phone up and say, well, could we have some product? Well, we can't just turn on the tap. It's 6 months to actually get from start to finish. One of the reasons why there's such a big loss is the QC sampling for stability studies. We have quite rigorous ICH compliant stability studies. So we end up with 548 ampules, but we only got 368. So we lose about 30% of what we make goes into QC. So that's why we've reduced the size of the vial from 10 mil to 4 mil, because in stability studies they need a complete sealed ampule or vial. So we need 88 because of this validation of the assays. So by switching that, we save nearly a litre of final product. So we've touched on this subject, and this comes into sort of health economics and the like. We make an Equitab G as an IgG. It takes 2 weeks to make. It's a lot cheaper. But in terms of Bothrofav, the regulators insist that we give the patient a safe product. The IgG product is safe, but it's very difficult to wind back the clock and say, well, yeah, we're going to give you an IgG. It's cheaper, but you may get some side effects. Now, Benjamin talked about side effects. We did a phase 1 study of an IgG product in healthy human volunteers, and the incidence there was no anaphylaxis, but the incidence of delayed serum sickness was 80%. So if you pre-med the patient, you can reduce that. But in a sort of proper, sort of controlled phase 1 study, the incidence of delayed serum sickness was 80%. So in conclusion, we've made a huge investment in the new manufacturing facility. We transferred quite a complicated process across from Sanofi, which we've had to downscale to make it appropriate for the volumes of products that we actually make. And really, between my core team, there's probably over 100 years of experience in immunoglobulin purification and manufacture. So it's a combination of a good quality system to ensure that we comply with the regulators, plus great technical expertise. So this little phrase is, or saying is for the clinicians here in the audience. Medicine is a science of uncertainty and an art of probability, but the safety and effectiveness of the tools we use should never be uncertain, and that's one of the big investments we make in ensuring that we do. So just a quick acknowledgement, to my team on the ground who actually make this possible. It was a long slog. Given how difficult it was where I started, if I knew what the journey was going to be like, maybe not, but you can't stop. And also acknowledge Sanofi, who gave quite patient and helpful in ensuring the successful outcome of the project. So that's it. Okay, thank you.
Thank you very much. Cameroon, for your presentation. Now the floor is open to everyone. If you get questions, I'm asking all my colleagues to come over. You can come over here, please.
Aaron.
Thank you very much to the speakers. I have a few questions for Ian. And one out of curiosity, this product you are developing from Posidoides difficile.
Yep.
When it goes through the gastric, to the stomach, doesn't it get disrupted by the acid pH?
It would do, but we've formulated it with antacids to get it through the stomach, and then with proteolytic enzymes, another excipient to prevent digestion by proteolytic enzymes in the small intestine. So yeah, so it works in hamsters, hamster stomachs are different to humans, so that's one of the things you'll find in the clinical trial. Does it really work in the human gut? Yeah.
I have two specific questions from the fractionation
Yeah.
process that you mentioned. One is that the chromatography uses ion exchange or affinities, ion exchange chromatography.
The first one is IG capture, and the second one is basically binding. It's the FC.
Another curiosity I have, how much do you lose when doing pasteurization?
That's a good question. We don't see much precipitate, but what we believe is, there is some denaturation of the antibodies, so perhaps you're losing some binding. But in terms of, because we obviously put it through a filter post pasteurization, but we get very little sort of solids coming out. So we think it's mostly denaturation.
Thank you. Congratulations for the presentation.
All right, thank you, Gemma.
Questions for Aaron, Atem Kalel. I have a question for you, Professor Kalel. Do you hear me? Okay. Can you, I don't know if you are planning to work on time 20 Venom 2? What can we expect for time 20 Venom 2? Maybe from 9 hours to 6?
Atem?
Do you have any question? Questions? We go in for 10 minutes, Jose? 10 minutes, coffee break? 10 minutes for the last session. And after you are free. Today is Friday. 10 minutes.
We also have 2 snakes in display outside, one feather lance and one boa for viewing.
to apologize for my English. I'm going to present a quick review of the process of the production of antivenoms. We have installed at the Instituto Plomero Picado in Costa Rica to produce antivenoms formulated with whole IgG, which are purified by the caprylic acid method. So, the first step in the production of antivenoms is the selection of venoms used as immunogens to stimulate the immune system of the animal selected as immunoglobulin source. We have selected horses. Thank you. We selected horses because they have a strong immune response, and they have a sedimentation of red blood cells. It's spontaneous. They do not need centrifugation. So there are many advantages of the use of horses. Horses are immunized with the snake venoms. The way to produce those venoms is by maintaining a collection of venoms, snake venoms in captivity, and then extract, milking the venom from the snakes. The venom must be active, antigenically representative of the group that they belong in, and the venom must be stable. With such venoms, you immunize animals. As we mentioned previously, horses are selected because they have docility, strong antibody responses, and high plasma productivity. And the immunization could be made with a single venom to produce monospecific antivenoms, or with a mixture of venoms to produce polyspecific antivenoms. The design of the process is a issue that must be carefully think. It could be taking account the number of venoms, the type of venoms, the dose of venoms, because you can kill the horses, and the frequency of administration, the way by which the venom will be administered, and the injection volume, for example. Another thing is the type of adjuvant, immunological adjuvant that will be used. Adjuvants are used to enhance the ability of snake venoms to stimulate the immune system of the horses. There are, when you produce polyspecific antivenoms, you can have cross immunomodulatory effects between venoms. Some venoms increase the strength of the response towards other venoms used as immunogens. And also, similarly, some venoms decrease, have immunosuppressive effects on the antibody responses toward other immunogens. So the mixtures must be selected and designed according to the properties of the venoms on the antibody response induced by the venoms used as co-immunogens. Other thing is that the venoms should produce the low damage in the horses as possible. And the effects of venoms in the horse can be local in the point of injection of the venom, and can be systemic. As you want to preserve the health of the horses, the immunization must cause the minimal of null damage during the immunization. As a result of injection of venoms, the immune system of the horse reacts producing antibodies. These antibodies are obtained by bleeding of the horses from the jugular vein. Normally, you take samples of blood. That blood, this is here it is, Dr. Labora, in an experiment we made to test the electrophysiological effect of bleeding on horses, just to be sure that the blood collection does not affect the health or the physical or the general status of the horses. And after rest the blood in refrigeration, the red blood cells sediment, the plasma is a supernatant, and that in such plasma is where the antibodies are dissolved. So the quality control of that plasma is very important, because bleeding happens in a very contaminated environment. So microbiological contamination of plasma is a easy thing that it is very probable to occur. And therefore, a very strict aseptical technique to take the bleeding is important. Anyway, just the plasma with the microbiological profile suitable for process is accepted, and all the plasma without that quality profile must be discarded. So as previously mentioned, antivenoms can be formulated with whole IgG or FAB2 fragments, which are obtained by pepsin digestion of equine antibodies. Anyway, the start material to produce antivenom is plasma. In our process, plasma is precipitated by the addition of caprylic acid during approximately 1 hour. And then diatomite, with the assistance of diatomite and deep filters, the bulky material that is precipitated is removed. And then we have a filtrate material that is basically is a solution of whole IgG in which other proteins are not present. This solution is dialysed to remove residual caprylic acid and other salts, and the solution is filtrated and then formulated with phenol, with sucrose, with excipients to prepare the formulation for filling. All this happens in just 1 day, from plasma to the bulk for filling. And when the quality of the bulk is verified, the product is filled in 10 milliliters vials, and then is stabilized by lyophilization. And then there is a process of cosmetic approval by an optical inspection of the vials, and the labeling, and all the tests of the final quality. There is an important step in the production, that is, during the clinical use of the antivenom, we need to know how is the performance of the antivenom in terms of efficacy and safety, and all the information we have regarding such performance is used to feedback the design of the antivenom and improve the quality of the antivenom. All the process it depends on all the steps of the process. For that, you need the most important resource is people People must know how do the work they do. And in the case of ICP, we have a lot of people with very ability to do each step of the antivenom. Quality is not product of causality. Quality must be produced during the process. And all these people, that is the work they do. And it's a quick presentation because most of these things were presented previously. And thank you very much.
Thank you so much, Dr. Guillermo. Now I invite Professor Frédéric Laposton for the next talk.
Thank you. So, I'm happy to be with you. Nothing with a snake. And the question was about France. So, you've got to come with me in France. This morning, my friend Papagaï explained the pre-hospital system with SAMU, the history, and this is the daily practice of emergency medical system in France. There is a unique phone number, which is 15, and something special, which is that in every case you call for an emergency problem, you will speak with an emergency physician. Final decision will be taken by an emergency physician. This emergency physician, for example, can give you just medical advice in order to avoid going to the hospital. He can send first aid responder, usually in France, fire brigade. He can organize a transfer to the emergency department by ambulance, for example. And in case of critical situation, he can send mobile intensive care units. The second point, which is specific in France, not absolutely, but is that in the mobile intensive care unit, you've got a physician. You've got an emergency physician, usually with a nurse, with a specific training in emergency medicine, a driver, and usually a student. The aim is to be able to do what we could do in the emergency department, including biology, for example, including antidotes and ultrasound, to manage the patient until the emergency department, or to switch emergency department to go directly to operating room or intensive care unit or cath lab in case of acute myocardial infarction, for example. The aim is to have the good patient at the good place at the good time. It means that sometimes you don't go to the hospital closest to the patient if you need immediately operating room, if you need immediately cath lab access. So this is a crucial point if we discuss about antidote therapy. Where do we have to go with the patient to be sure that antidote will be immediately available? First point is this one. Antidotes have to be immediately available for all the patients. Many years ago, we wrote this paper to say that it's really part of the mission of the SAMU to have organization allowing every patient, everywhere, at every time, to have, as early as possible, antidote therapy. More recently, this point of view has been supported by this paper, where the hospital, as you can see, had some problem just because antidote was not available 24 hours a day. in this case, it's interesting to see that it was digoxin antidote because it's a kind of antidote we don't use. The use is not daily. So the problem is to have a stock of antidote that maybe you won't have to use. The second point is this one. What kind of antidote do we have to stock in each hospital or in each emergency department? And this question is asked regularly, but it's really difficult to answer, to have a definitive answer. The point is probably the third one. Some antidotes have to be immediately available, and then in French system they will be in the ambulance, and in other cases we can wait 1 or 2 hours. The first list for France has been published 25 years ago with these antidotes. They are supposed to be in every mobile intensive care unit in order to be infused immediately, and thus in 2 hours. You see, there is no antivenom in these 2 lists. Update of this list, more recent, and it's in French, but you can translate that. For the first time, we see in the list of antidotes that we are supposed to be able to infuse in the hospital, antivenom is appearing recently. This is the transition with the first part of the presentation, which was snakebite in France. Even if we are in the zone where we are supposed to have snakes, most of French snakes are not in France, not exactly in France, in overseas territories. We saw Guyana first. We don't speak about New Caledonia second, Martinique, Guyana first, Martinique also. But we do have some exotic snake bites in Paris because it's more and more popular to have exotic animals at home. Just before coming, I tried to buy one, and you can see that it's not so expensive. The problem is that sometimes you can have accidents, and usually you will see the reports in the newspaper. You also know that Paris is not yet an exotic city. So we don't have any snake in Paris. And in comparison with what you spoke about Asia or Latin America or Caribbean, you see that the rate of snakebite and envenomation in France is low. Just to give you an idea, we don't have statistics because, you see, we don't need very precise statistics. In France, 65 million inhabitants, 2,000 snake bites every year. Only 500 envenomation and 1 death. Unlucky. You could say that, and you're right, it's not a problem in France. Yes, but on the other hand, you can ask, we've got a very organized system for emergency management. There is still 1 death. And the second point is that if you don't use to routinely manage this kind of patient, clearly you increase the risk of mistake. So we should not consider that it's not a problem in France. Because, back to this slide, we've got, and it's here, we've got 4 snakes, all vipera. And you see that we could say that there is only 1, because for these 2, it's, only very small territories. So in most of the cases, Aspic is responsible for envenomation in France. I don't speak about clinical feature of the patient. I don't speak about antivenom because it will be done by Bruno Megarban just after. But as I told you, we've got physician in pre-hospital setting. The question is what to do before reaching the hospital. We can start the treatments. We can start fluid resuscitation if necessary. We can start the use of painkillers. So it means that symptomatic treatment has to be started as early as possible in France in pre-hospital setting. The question is also the location. It's not possible to have all the antidotes in all the hospitals in France and in every country. It's too expensive. And if you think about antidote for digoxin poisoning, you need it. But you cannot have them in every hospital. It's not possible. The relative question is how to carry the antidote to the patient. In France, one of the problems is that the antidotes are not always at the same place. Somewhere with the ambulances, with the mobile intensive care units, in the ambulances or at the station. Sometime in the ED. Sometime in the pharmacy of the hospital. Sometime in the intensive care unit. Sometime in the poison center. It depends on the organization of the hospital and also of who is involved in the management of poison patients. The problem is to be sure that in every case, regardless of the location of the antidotes, you've got the list of the antidotes, indication, and the way to use them. You are sure that they are available 24 hours a day. It means that someone in the pharmacy is able to take the antidotes, and it's not always the case in the hospital. And you are organized to bring the antidote to the patient. For example, in my area, this is the call center in the middle of the department. 4 big hospitals, few small hospitals, and mobile intensive care units in big hospitals. The stock of antidotes is in the academic hospital in the middle of the department. We can be called directly by the patient, but also by emergency physician in the emergency department or in the intensive care unit of one of these hospitals. And then we are responsible for the discussion about the indication of antidotes and the transport of the antidote to the patient in the ED or in the intensive care unit or sometime at home. But in my region, you see, it's close to Paris, and as there is no snake known to live in Paris, we don't have any antivenom in my department. This organization is the same that very specific strategy in case of poisoning with ECMO. You cannot, at 2 o'clock in the night, decide how to organize access to ECMO. It's exactly the same for antidotes. If you've got at 2 o'clock in the morning to find an antidote, you will fail. You have to be organized previously. Just to finish, 2 or 3 points. Due to the kind of snake we've got in France, the target is the legs. Quasi exclusively. You spoke this morning about season. You see that in Europe, there is at least 6 months without any risk of snake bites. You spoke about that yesterday. We've got a lot of dogs in France, of domestic dogs. and cats, and so on. And they are also responsible for many envenomation. Due to climate changes, and deforestation, the population of snake is changing in Europe. And we don't know, you spoke about that yesterday, we don't know what the population of snake will be in Europe in 10 or 20 years. We don't know. Some of them are decreasing, others are increasing, so we don't know. And there is something which we could say it's funny because we've got venomous and non-venomous snake, and you will find many places to discuss to be able to distinguish venomous and non-venomous snake in France. And you spoke about yesterday, maybe you could be helped by your phone. Okay, I'm not absolutely sure that this is a good strategy. The best strategy probably will be to avoid the contact with the snake. In some countries, it's famous to have alcohol with a snake. We don't need that in France, of course. We've got good wine. And you know that France is the country of fashion and cosmetic. And we use snake not for the wine, but for cosmetic. Maybe it will be better with more snake in France. Maybe. Okay. We are going on with the clinical feature of French snakebite. Thank you.
Thank you very much. So we will be staying in France a little bit longer. I'm not from Jamaica. And our next speaker, if we were lined up, will be Professor Bruno Lajban. Did I pronounce that correctly? Professor Bruno Ligaba, the Chief of Department of Medical and Clinical Toxicology at Lariboisiere Hospital in Paris. Thank you.
Yes, we hear you.
Okay, perfect. Sorry for not being with you. I'm very happy to give this short lecture about the management of snake bite envenomation in France. So unfortunately, I was unable to join and come to St. Lucia because I am currently in the UK, and I just arrived in Glasgow because of a delay in the flight. So I'm in the airport, and sorry if you are hearing some music because I'm in a coffee shop, and hope the presentation will be okay. So please forgive me for any approximation because the conditions are not so good. So I have no conflict of interest.
Hello, Professor Megarban. Can you speak louder, please?
Yes. Do you hear me? I cannot speak louder.
It's better.
It's better. Okay. I have my head just stick to the screen. So sorry. I cannot do better. So I have no conflict of interest to declare with this presentation. As you know, viper bites in metropolitan France are primarily caused by two species, two kinds of vipera, Vipera aspis and Vipera berris. And you see on the slide their repartition, their geographical repartition in France. There is also a seasonal distribution, and you see that the majority of bites occur during summer, between April and September. We have, as said by Professor Lapostole, about 2,000 cases of snake bites and about 500 cases of envenomation in France each year. So antivenom therapy plays a crucial role in mitigating the systemic effects of the venom, even in our cases in relation to Vipera bite. So in France, this slide shows you the epidemiology of the patients bitten by vipers. They are mainly males, adults. Bites occur accidentally and sometimes, rarely, due to manipulation. They are responsible for hospital admission with short length of hospital stay, less than 24 hours in 38%, and 2 days in the majority of the situation. So generally, the envenomation is not so severe. However, they regularly require the administration of antivenom, and you see that in this series, about 94% of the patients received the antivenom, and of course, some part of them will receive antibiotics, anticoagulants, and exceptionally corticosteroids. These envenomation are mainly responsible for skin edema, but in some cases there will be some biological impairments, mainly an increase in blood white cells count, a decrease in platelet counts, and a coagulopathy with an increase in the ENR, a decrease in fibrinogen, and the appearance of D-dimer and products of fibrin degradation. There is an envenoming scoring that has been assessed in the 90s, and you see there are 4 grades, one with no envenoming and grade 1 with minimal envenoming with local edema, grade 2 moderate envenoming with regional edema, and grade 3 with severe envenoming with extensive edema, and of course systemic impairments, hypotension, shock, necrosis, and organ failure. And you see that there is a quite good correlation between the amount of circulating venom, the concentration in blood and even in urine, and the grade of envenoming with viper bites in France. This slide shows you the usual clinical presentation in France, and you see that less than 5% of the patients will present severe envenomation. In the majority of the cases, there is mainly minimal or moderate signs, and mainly local or regional edema. In some patients, however, there may be some systemic signs with hypotension, shock, severe pain and ecchymosis, and some anaphylactic or hypersensitivity reactions. Of course, coagulopathy is a major complication and will be observed in the most severe cases. More recently, a new VIP grade scale was determined for improving the medical management and the antivenom administration to the patients in case of envenomation following the snake bite by the vipers in France. And you see this scoring is based on 5 grades, and the grade 2 was divided in A and B to encourage more largely the administration of antivenom to improve the management. And this is now validated and is also available as computerized clinical decision-making tool to assess the severity and to allow a better management and follow-up. So antivenom therapy is available in France, is largely available as explained by Professor Lapostole. And the main compound is called Viperfab. It is a lyophilized equine serum-based formulation containing polyvalent FAB prim2 fractions of purified immunoglobulin produced specifically against European Viper venoms, including Vipera aspis, Vipera berus, and Vipera ammonides. The standard dose is to infuse 1 vial in 250 ml of saline or glucose over 30 to 60 minutes. Of course, dose adjustment is recommended based on the clinical response, and a continuous monitoring of the patient is recommended in hospitalization because of the risk in some patients of allergic reactions. Of course, the conditions of administration of this antivenom were improved since it was guided by the severity scoring systems. These antibodies, these FAB prim2 fragments, were shown to be safe. And of course, it was assessed that early administration will result in a rapid improvement in the patients. Of course, in France, due to the rare cases of severity, expected outcomes of administrating this antivenom cannot be relied on the decrease of mortality, but rather on other scores. With this antivenom, the rapid neutralization of the venom components will result in the decrease of morbidities and mortalities. That mean the prevention or the treatment, the rapid treatment of systemic symptoms such as the coagulopathy and shock, and of course the prevention of long-term complications due to tissue necrosis, which may result in an improved quality of life. And of course, there is some issues regarding the cost effectiveness of this therapy. It was demonstrated that Viperfab administration is associated with a shorter hospital stay and a quicker recovery. Indications are relatively large, and the specialists in France recommend to administer this antivenom therapy earlier and in moderate to severe envenomation. That means grade 2 or grade 3A and grade 3. There is, of course, indications that will be modified by the monitoring of the injuries. A rapid progression of the local symptoms with severe swelling and blistering could result in the administration of the therapy. Of course, systemic symptoms, including hypertension, coagulopathy, neurological effects, will be a requested indication. Of course, in some vulnerable populations, including children, there may be extended indications to avoid severe complication in this population. And of course, anyway, the clinical decision will be based on the severity scoring systems and the monitoring of the patients. The Viperfab is largely available in France based on the poison control centers, and they are available in all the regions, especially in the south of France, where there is a higher risk of envenomation. Of course, they may a shortage in the antidotes may occur, and there are two alternatives. Recently, Viperfab was not available, and Viper Atab, produced in the UK, was used. However, as you see, these are FAB of specific immunoglobulin for Vipera berus, but not for Vipera aspis. And it may not be successful in these envenomation. Thus, some antivenoms specific for Vipera aspis were obtained from Italy. However, imported antivenoms require special authorizations from health authorities, and this is not a relatively easy process, especially that the administration is usually emergent. So just to conclude rapidly, and sorry again for the conditions of this presentation, of course, envenomation with vipers in France is relatively rare. Just 500 cases are reported yearly. There are two major vipers that are responsible for envenomation. Anyway, rapid assessment is necessary to reduce complications. Antivenom should be used according to the severity. Specialized advices should be collected from the poison control centers. And of course, anticipation to avoid shortage in the seasons in the summer where the bites occur is important. And today in France, hopefully, there is enough available antidotes in almost all the regions to care about the patients and to avoid any case of death. Thank you very much.
Thank you very much, Professor. And our next presenter is well known to us, Dr. Evan Gomez, who will be speaking this afternoon to us about the new antivenom that are available in the Caribbean region. Thank you.
Clicker.
You can do clicker with Zoom. Zoom don't allow it. Zoom said no.
Okay. 10 minutes. Can I do it in 10 minutes?
You can do it even more fast. Okay. 10 minutes is good.
I'll try to do that. Good afternoon, guys. Again, I will try to be very quick because just to wrap up the today's session, because we have some snakes outside and it's Friday. Yeah, considering that. For the last talk, I would like to just to take you guys and walk with me in this kind of a small, tiny story we are about to tell you. So I just came up with this title, which is could be a new antivenom for the Caribbean region, the Polyval ICP Caribe. Where's my French professor? Caribe. Thank you. So again, some of the topics. So brief history. We were been talking about this pretty much the whole day. Some immunization strategies. Dr. Memo already mentioned the strategies that we used back at the ICP in Costa Rica. So I will just flying by through those. The project's timeline, which is the history itself. Try to just walk with me. Some of the major results. Just don't play with me, man. How about okay. See, I'm very fancy now. So some of the major results and some of the conclusions just to end up the session. So just to resume and briefly, efforts have been conducted to develop the national and regional efforts to confront the neglected tropical disease. We already know that. The epidemiology of snake bites in the region is largely unknown, and we have been talking about this, and there is no systematic register of the neglected tropical disease, or is available. So there is a lot of under records still and that definitely impacts the whole snakebite situation in the region. And snakebites, as neglected tropical disease, have not been considered as a priority issue approach in the Caribbean health systems. So, okay, let's try to do this very interesting history. So we heard from Dr. Dabor, Professor Dabor, yesterday, the beginning of all this collaboration with the South-South and Global South. So at some point, he reached out to Dr. Gutierrez, also known as Chema, and at some point, I don't know how I just got involved. So after several conversations and some meetings and stuff, by 2022, I just end up in CHU Martinique Hospital, doing crazy things as collecting venom from fertilizers there. So that was the start of the collaboration when it comes from the ICP itself producing the anti-venom, the new anti-venom for the region. So in 2022, we did the first venom extractions. That's me again doing crazy things, don't pay attention. But that's the result of that journey. So we have the first, I would like to say, the first batch of Botrops lanceolatus from the Martinique island is as the liquid form and then the lyophilized form. That was a very good effort from Professor Dabor and his team to collect all the equipment, the resources to have the venom to start off this new journey of trying to produce a new anti-venom for the region. So as Dr. Memo already mentioned, we have some strategies, and the idea behind this was not to come up with new things, not to reinvent the wheel and stuff. It's just to apply that we already have settled and functioning very well to this new component. So we start off with 4 horses as an experimental group, and adding the thing that we like to call now the Caribbean venom component, the CVC, which is conformed by Botrops atrox from French Guiana and Lanceolatus from Martinique. So every time you are going to see CVC, we are adding this component to the already established mixture of antigens or immunogens or otherwise venom. And the main idea was just to implement and add it to the routinary immunization schemes. So we are not doing any crazy new things. If working, don't fix it, or kind of some sort of the phrase in English, sorry. And we use specific adjuvants. Already, Dr. Memo mentioned it. We kind of have tested out several adjuvants. In this case, particularly, we use montanine, which is very, very good, minor to pretty much none of the side effects, let's say that, and produces or stimulates a very good response in, let's say, in like short middle time. This is the one thing that I will take your attention during the whole presentation. So let's try to begin with the story. So in 2022, we started collaborations. We did the first Botrops lanceolatus venom extraction and providing the initial step for new antivenom. Again, if we don't have the venom, we cannot go up and escalate all the process. So we need one of the main, raw resources to work with, which in this case is the venom. By March 23, so this is almost 1 year, we do the pilot project with 4 horses and adding the CVC component to the Viper polyvalent mix. So you is just adding, at this stage, we already had the Botrops component, which is Botrops asper from Ecuador, and we had the Crotali component, which is Crotalus simus from Central America, which is Costa Rica, the tropical rattlesnake, and we add a new component from another rattlesnake, which is a crotoxin-like, which is Crotalus durisus pifanurum from South America, specifically Venezuela. So that thing is a different story, but it's very important because you will see some points that just stimulate some of the responses. And of course, we have the lachetic component, which is Bushmaster lachesis estenophrys. So again, we did, I think within the 2022, we did at least 3 batches of venom that we already have and start work with. And by September 23, we just need to keep up and trace and have track of all the things that actually all the immunizations that we have been doing. So we did a couple of things. We kind of end the pilot immunization scheme and perform some neutralization test, like how you call that in English? It is a phishing? No. It's late, sorry. Effective dose, meaning effective dose. Thank you. Don't blame me for that. And some ELISAs just to follow up the behavior, let's say that of the plasma pools. And these are results, some of the results that we have. So if you can see here is before the immunization and after the immunization of raw plasma. And this is the important part for the first batch and pilot group. So we have some of the boosters. So we have the Caribbean booster here, which is are the red arrows. So you can see that we have some good, let's say good because they were hyper stimulated before, but when we added the CVC and start to see some of the increases of the responses in terms of antibody titers, will be interesting to keep this trending in increasing the response. At some point we did the Crotalus booster, and you can see there is again another response or increase in the response, which is very interesting because Crotalus is a different, let's say, actually it's a different genus, same family, different genus, but it's stimulating the whole response of the group that we use. And this is for Botrops atrox as well. So you can see it's pretty much the same trend of having a good response after the 4 boosters and then with the Crotalic. So those were the very promising results. So we just continue to February from this year. We add the CVC to routinary immunization scheme. Now we just scale up to 24 horses. And as we call the polyvalent group 1, so we have already one group on the industrial process, just being I can't speak English today, sorry. Being immunizing with the CVC component. By March of 2024, we just continue with the routine of the immunization, and we have been applied 4 boosters of the CVC, and again we did the effective dose to the plasma pools, and those are the results. So as you can see, all right. So you can see we did the intraperitoneal lethality dose and then the effective dose. So you can see for lanceolatus, I'm just rushing up because of the time. For lanceolatus, we have some, let's say, mild response, and then we just see the increase in the antibody response. Remember that those are the raw pools. Sorry for this one, it's still in Spanish. See? Now I'm late. And as you can see, those are the species that we are using in the scheme. So for Botrus asper atrox and lanceolatus, we are seeing that we still have like a but it still have a good response in terms of just continues with a hyperimmunization of the horses that we add into the industrial process. By May of 2024, we expanded to the whole, let's say, to the whole herd of horses. And we now are talking about 33 to 70 horses that were used to be immunized with the CVC component. And by September 2024, we already testing several batches of the Polyval ICP Caribs. Again, all the physical, chemical, all the quality control, and all the quality set that the Dr. Memo already mentioned, we are applying to these batches. So we have at least 5 batches of Polyval ICP Caribs. So this is one of the first, so it's just one group, and then the addition of the second group as just scaling up to the industrial. This is the same, just moving here to the entire group and to the entire herd. So you can see the pools up to October 2024, and this is the final product, which are the batches with all these, well, as I already mentioned, with some of the biochemical features that are important as protein content and effective dose. So we have all these 5 batches, which are behaving very, very good compared to Remember that we are a polyvalent, just trying to targeting as much as the species that we can consider in immunization without affecting the welfare of the animals. In this case, the animal model that we're using are horses. And the major results, for just to conclude, as we were able to manufacture at least 5 batches of the CVC, polyval ICP. So we have proven neutralization effectiveness and with safety protocols, and we have a well-established snake species coverage. But again, remember that I show you some of the fluctuations in terms of developing and try to stay hyper immunize the horses. So the conclusions for this are just adding the CVC to the routine immunization scheme show an increasing in neutralization effectiveness. However, some fluctuations in plasma antibody titers were observed, and in order to keep these high plasma antibody titers, horses must be challenged at least every 2 months. That means that we require venom supplies regularly. And some of the next steps, just to conclude, is strengthening and consolidating global South collaborations and decisions, which is kind of the main focus on this, have to be made by stakeholders parties. Again, all this made thanks to all these people in the ICP antivenom team and all the sponsors. Thank you, guys.
Thank you, Professor Aaron. Now I invite you, please, Dr. Guillermo, Dr. Frederic, and Dr. Aaron to join us here in the table for possible questions from the audience. I think Professor Bruno is online with us, yeah?
Yes.
Any question? Dr. Gutierrez?
Thank you very much for the presentations. I have a question regarding the treatment of envenomings in France in terms of whether there is a protocol also for first, whether you have bites by exotic snakes, and how do you handle these envenomings by probably very different exotic snakes in France?
You're right. This is one of the problems, but by chance or not, only a small number of accidents. And in my experience, I had in my geographic area no case. But maybe Bruno, il est plus en ligne. I don't have any experience of this exotic, unexpected snake in France. And I don't see any publication about these specific cases. But it's probably not specific of France. It's specific of a non-exotic area where you want to have exotic animals.
Anybody else? Other questions? No? Maybe anesthesiology practitioner have any question? No?
It's Friday.
It's Friday, yes.
We arrived yesterday, but we already know the rule. It's Friday.
Yeah. Labor? Where is Labor?
It's Friday.
Thank you so much for your presentations. Thank you, Doctor, for your company and your table.